Let's introduce ourselves with the structure of DNA. This is the basic preliminary description of the structure of DNA. In my next video, you will find the most appropriate model of DNA or the structure of DNA, which is Watson and Crick model of DNA. So to begin with, it is important to know chromosomes. What are chromosomes and how it is made up of? Chromosomes are made up of nucleoproteins, which constitute base proteins and nucleic acid. So chromosomes are made up of proteins plus nucleic acid. Na stands for nucleic acid. Two kinds of nucleic acids are there, DNA and RNA. DNA stands for deoxyribose nucleic acid, whereas RNA stands for ribose nucleic acid. Sir Friedrich Mescher in 1869 first reported nucleic acids from the nuclei of the past cells. So he found nucleic acid from past cells. He stated that the nucleus of cells contains some acidic substance, and that acidic substance is termed as DNA and called them as nuclein. In cytoplasm, these are found to be associated with protein synthesis, because in central dogma of molecular biology it states that DNA transcribes to RNA and RNA then translates to produce protein. So this is the central dogma of molecular biology. So before that, DNA replicates itself to process the rest of the process, which is transcription and translation. So in cytoplasm, these DNAs are found to be associated with protein synthesis by a process called translation, by forming the intermediate mRNA or messenger RNA. DNA is thus considered as the storehouse of genetic information or the genetic material, and transmits information to the offsprings. Number of nucleotides determines the length of DNA. So how many nucleotides are present, that determines the length of DNA. Now to say that what are nucleotides, these are all new to you. I'll explain this in my next video, which is the Watson and Crick model of DNA. Nucleotide is actually sugar plus phosphate and any types of purine or pyrimidine, that is either any one, adenine, guanine, cytosine or thiamine. So these are all new to you. You should remember by now that number of nucleotides determine the length of DNA. Now base pairs, pair of nucleotides. Bacteriophage has 5386 nucleotides, and lambda bacteriophage has 48,502 base pairs. Most common gram-negative bacteria, Escherichia coli, E. coli, has 4.6 into 10 to the power of 6 base pairs. BP stands for base pairs of nucleotides. Now what are base pairs? That is a pair of nucleotides. I'll all discuss this in the Watson and the Crick model. This is just the introduction to the structure of DNA, so you should remember. A haploid set of human DNA has 3.3 into 10 to the power of 9 base pairs. Now the components of nucleic acids. DNA and RNA contains pentose sugar, that is 5 carbon sugar, and the nitrogenous base. This is a sugar moiety or the molecule. This is the basic diagram of the sugar molecule or a moiety. So DNA and RNA contains pentose sugar and the nitrogenous base. Nitrogenous bases can be adenine, guanine, cytosine or thymine. Now what are nucleotides? Nucleotides in DNA are called deoxyribose nucleotides, which is made up of deoxyribose sugar, nitrogenous base and phosphoric acid. Nitrogenous base is again of two types, purines or pyrimidines. So in short we can say nucleotide is formed of sugar, base and phosphoric acid, which is a nitrogenous base, and nucleocytes are formed by a purine or pyrimidine, nitrogenous base and pentose sugar, but it is devoid of phosphoric acid, that is no phosphoric acid is present. So when phosphoric acid is there or phosphor is there, it forms the nucleotide. So this is the basic component of DNA or RNA. So in describing elaborately, nucleotides can be either purines or pyrimidines plus pentose sugar plus phosphate. Deoxyribose sugar. In DNA, deoxyribose sugar is present, whereas in RNA, ribose sugar is present. Now why deoxyribose is named so and why ribose is named so, I will explain in the next videos. So nitrogenous bases, these are of two types. So we confirm that nucleotide is formed of sugar, nitrogenous base, that is NB, and phosphate, so to say. So this nitrogenous base is again divided into two types. One is purine and pyrimidines. Purines comprises of adenine and guanine, and these are dicyclic moieties, whereas pyrimidines includes thiamine and cytosine. But in RNA, in place of thiamine, uracil is present. Okay, and pyrimidines are monocyclic, that is, it is single ring structure, and pyrimidines are double ring structures. Okay. Now nucleocytes, as I mentioned earlier, it is made up of nitrogenous base and pentose sugar, but phosphate is not there in nucleoside. Nitrogenous base is linked to the pentose sugar through a N-glycosidic linkage. This is important. So nitrogenous base, which is either purines or pyrimidines, purines comprises of adenine, guanine, and pyrimidine comprises of cytosine and thiamine, is linked with the sugar of the nucleotide through a N-glycosidic linkage. DNA nucleosides are known as deoxyribonucleosides. Phosphate group, when linked with the 5' hydroxyl OH of a nucleoside through a phosphate, so phosphate ester linkage, a corresponding nucleotide is formed. So nucleotide, as I mentioned, it is made up of nitrogenous base, which is joined with the sugar by N-glycosidic linkage, and phosphate group is also present, which linked with the 5' OH of the nucleoside through a phosphate ester linkage. Now these are very uncommon to you. I'll explain this in the Watson and the Crick model of DNA. So let me explain, like nitrogenous base, which is either purines or pyrimidine or pyrimidine. So nitrogenous base plus sugar forms nucleoside. Okay. Now when attaches to phosphate group, it becomes nucleotide. So this phosphate group is linked with this nucleoside with the 5' wedge of this nucleoside through a phosphoester linkage. Now nucleotides are linked through 3' 5, this is prime actually. Nucleotides are linked through 3', 5' phosphodiester linkage to form dinucleotides. Now Chargops law. After the name of the scientist Sir Erwin Chargaff, he measured that the quantity of bases in DNA, that is nitrogenous bases, the amount, that is the quantity, he measured that and noticed that quantity of adenine is equal to the quantity of the thymine in DNA, and quantity of guanine and cytosine are equal. So A plus T is equal to T plus C. This is important. So the number of adenine is equal to the number of thymine in DNA. Number of cytosine is equal to the number of guanine, and A plus T is equal to T plus C.